Hi, I'm Hamish.
I'm Peggy.
I'm Storm.
And kia ora, I'm Rose.
I intend on doing honours with the Biochemistry department and would one day love to go into research. Speaking of, we have a couple of very cool examples of research that we would love to tell you about. So our first example of research in the Biochemistry department at Otago is Lucia Speitz's research into Batten Disease. So what is Batten Disease? Batten Disease is a fatal neurodegenerative childhood disorder, and it's caused by a mutation in the lysosome, which means that the lysosomes don't function as they could, and this severely affects neuron function. Here's our lysosome. Symptoms of Batten disease include blindness, loss of motor control, and epilepsy.
So as we can see up on the slides over here, in our cell we have some of our most favourite friends. We have our nucleus with all our DNA, and we have our mitochondria making all our energy, and we also have a couple of lysosomes on there. So to explain what lysosomes are, put simply, they are the cell's recycling bins. And so I want everybody to take a second. We're going to do a little exercise. I want you all to think for a little second and now imagine that we are all in a cell. This is no longer a level 2 biology classroom. This is a cell. We're all hanging out at 37 degrees Celsius. We're having a really great time. And as we can see, here is our lysosome. Now, what I want you also to picture is when you get to the end of this year and you've finished up all your exams and you're really sad because it's summer and you can't do biology again until next year, what you're going to do is you're going to take up all of your level 2 biology notes, you're going to screw them up, and you're going to chuck them into the recycling bin. And what's going to happen is those notes are going to get broken down and they're going to get remade into something new. So that is exactly what lysosomes are doing in your cell. As the cell is going about its day-to-day function, we get a build-up of things like proteins and lipids and sugars, which you can see on the slide. They accumulate over time, and we have them in the cell because they're useful, but then over time they might get damaged or we don't need them anymore. So they get moved right into the lysosome. And as I said, the lysosome contains a bunch of enzymes, and these degrade everything and break everything down. These are, lysosomes, enzymes. And what happens is when they chew everything up, this gets spat back out into the cell, and those little degraded bits can then be remade into something new. And there we go. That is what lysosomes are doing. So what happens when they don't work?
Cool. So as you've seen, the lysosome is meant to work by rubbish going in, it's recycled, and then you get the useful bits back out. So a common reason that a lysosome will stop working is because of a mutation, which means the enzymes inside also stop working. So does anyone have any idea about what happens in the cell when the lysosome stops working? Nope, so that's all good. So the easiest way to explain to you is to show you. So if you guys want to do some recycling for us, chuck her at the lysosome. You guys are all good. So as you can see, the lysosome not working is the equivalent to us taking away your class recycling bin. So first of all, there's no more recycling. So there's no new stuff being made. And secondly, as you can imagine, if we kept this up, you would get a huge accumulation of rubbish in the cell. And this is really bad for the cell. This generates an awful toxic environment and does result in cell death. In the case of Batten disease, fully functioning lysosomes are so important for neuron health. And in Batten disease, it's actually a mutation that stops the enzymes inside the lysosome working, which causes a buildup of rubbish in the neurons that results in neuron death. And neurons, unlike our other cells, do not regenerate. And it's for that reason that Batten disease is fatal. Now that's some pretty heavy news and that's pretty morbid, but don't worry, Rose has some good news for us.
Okay, so as we mentioned earlier, Lucia at the University of Otago is doing some very significant research. And the reason it's so significant is because her and her lab group are trying to develop a cure for Batten disease. And currently there is no cure, there is no forever fix for this disease. And as we said, it is fatal. So very, very important work that she's doing. One experiment that her lab group has done is they took a bunch of sheep which had mutations in the genes needed to encode these enzymes. So therefore, these sheep, they didn't have functioning lysosomes, they essentially had Batten disease. And what they did is they injected these sheep with the genes encoding those enzymes in the hopes that then they would start to produce those enzymes. And that is exactly what happened. The sheep started to produce the enzymes they needed in their lysosomes. The neurons, were functioning, they were healthy. There we go. As we can see, we're getting our enzymes back into our lysosome. This is exactly what was happening in the sheep. And what they found is these sheep remained healthy. They lived a lot longer than what was expected of them if they had Batten disease with no treatment. They didn't degenerate, and they didn't have all those terrible symptoms of Batten disease. So this was very, very promising research. And now what Lucia is doing is building off of what they found and looking at neurons very specifically. She actually grows them in petri dishes, which is really cool, and trying to figure out a way to cure Batten disease in people. And I had a little look last night, and I actually found that within the last month, this gene therapy here that they did in sheep has actually been approved for trials in people and children that have Batten disease. So that's like the main message I want to get across, is that this is really groundbreaking, and it's been described as a world first gene therapy for Batten disease. And that's the really cool thing about biochemistry, is like we're using our knowledge, as you guys know, about gene expression. That's what this is. We're using our knowledge about gene expression to help people that need it. So after that, we thought we would have a little bit of a brain break and have a little play around with some protein structures. So over here, I have a 3D model of this protein called CLN5. And this is actually one of the enzymes in your lysosomes that breaks things down. And you can see over here we've got some sugars attached as well. And I thought that you guys could pass this around and just have a little look at it, because it's pretty cool.
So this is phycoerythrin here. It's a protein that gives the red pigment in red algae. So this here is subunit A, and this one's subunit B, and it kind of goes like to pass around. Just like, this is another way of displaying proteins. Just
So the proteins that have been passed around to you there, they are obviously the 3D printed models, but they've been produced through observational techniques, because proteins are small. It's not like we can just take a picture of them, so we have to turn to other techniques to get images of them. And those techniques were called electron microscopy, or for example. So when we do this, we get an image generated, and we can use computer software to observe it. And I'd hope to have it up on the screen to show you, but for a bit of a technical glitch we can't, sorry. So if you can strain your eyes, I'll try and show it to you here. Can everyone see that okay?
Yes.
So this here is a program called PyMol, and it's a computer program that we use as biochemists to observe the structure of proteins once they've been generated through those techniques I mentioned before. And this one here, this protein, is called the voltage-gated sodium channel. So what this protein does is it's the thing that makes your nerves go. So you think of the electrical signal travelling through your nerves. So this is a protein that does it. Firstly, you can see all of these twisty things. Can you manage to make them out?
The white.
Yeah, the wee grey twisty things. So those are known as alpha helices, and they form the structure of most proteins in your body. And they go through the cell long ways, because they provide a environment that they're stable and sitting in there. The other thing I wanted to point out to you is if we observe from the top, sorry, just let me find it, we can observe there's a hole in the middle where my mouse is. I know it's a bit hard to see, but that pore there is actually where the sodium ions flow through. So they go from the inside to the outside of the cell, and they conduct that, the electric signal on. And that relates back to the main point of why looking at the protein is useful, is that the structure of a protein relates to its function. So if we look at the protein and we can see it has a hole in it, or it has a big flat surface, we can look at it and think, oh, that could be a binding domain. That could be where a ligand joins to it, or it could be where something passes through it, like in the case of that.
We are currently living in a climate crisis. Global warming is evident in our oceans, with the seawater temperatures rising, leading to acidity and decrease in oxygen level. Ocean acidification decreases carbonate levels in our water. Young mollusks require carbonate to build their shells, as their shells are made up of calcium carbonate. Ocean's acidification due to global warming makes it harder for young mollusks to build their shells. The shell development is crucial because the larval stage is when they are most vulnerable. Growing shells quickly allows mollusks to be protected from the changing environmental stresses and allows them to survive. It has been found that some populations of the green-lipped mussels can grow their shells faster than others. This means they have a survival advantage. The focus of Nathan's research is finding out how ocean acidification and thermal stress affect young mollusks. He is looking at the green-lipped mussels, the classic ones you find at the supermarket. This research is crucial because of global warming threatening this species. So how is Nathan doing this research? He is looking at genes and gene expressions. Specifically, he's trying to identify which genes are involved in growing shells and how the expression of these differs between the mollusks that grow their shells faster and those that don't. For example, maybe the fast shell-growing mussels express these genes into proteins earlier. This enables us to understand the impacts of climate change on animal development and how species respond to survive in their environment.
Nathan is achieving this by using a technique called single-cell sequencing. So I'm sure you guys have all heard of the genetic code and how that's done, but what you might not have known is that we actually do have machines that do it for us, and they're really quite small. I've got a couple of examples up there. So that one's about as big as a thumb drive here, and the other one is probably a couple of those boxes over there. So they're really quite small. They're not taking up a whole room or anything. But single-cell sequencing, as it's so creatively named, is getting the genetic sequence of an organism by using one cell from it. And this is quite useful because it allows us to trace back where in the organism cells came from, and it gives us insight into what genetic variation they might have between the different organisms. So Nathan's research has done this, and it's shown that individuals have mutations present, not necessarily within protein coding genes, but within regulatory genes that affect how they grow their shells. So single cell sequencing, and you can think of it like this. So we've got some fruit bursts here that we're going to use for an example. So each bag of those fruit bursts represents a mussel. So when we sequence them using traditional methods, everything all kind of gets jumbled up together, and we don't know what really came from where. So we'll pass that around, and feel free to take a couple of fruit bursts. There's not many of you here.
Which you'll eat after class.
Yeah, after class.
After class, yeah, of course. So the thing with the single cell sequencing, with the regular traditional sequencing techniques, is within that bowl of fruit bursts is we don't know what original bag that came from. We can't track it back. But when the single cell sequencing technique is used, we can track it back to the original organism and what cell specifically from that organism the DNA came from. So you can think, oh well, a red one might have come from this mussel's heart, or the green one could have come from this mussel's shell membrane.
We all got into biochemistry a little bit differently, but we've all enjoyed studying it so much, and hope that at the end of this presentation you'll have a better understanding of what biochemistry is, the cool research going on in the department, and what it's like to study it at Otago. And to end things on a really cheesy note, if you love biology and you're fascinated by how things work and you want to make a difference, then we'd really encourage you to consider studying biochemistry. You might just be surprised.
Thank you. Yeah, thank you. Thank you. Thanks for having us.